Okay, I want to introduce our next speaker. Dr. Michael West is the founder and CEO of Ajax Therapeutics. He's focused his academic and business career on the application of developmental biology to age-related degenerative diseases. He was the founder and first CEO of Geron Corporation from 1992 to 1998. And from 1998 to 2007, he held positions as CEO, President, and Chief Scientific Officer at Advanced Cell Technology, Inc. And I just want to say, I'm very happy to have Dr. West back at RAADfest. As you can tell from his bio, he's one of the guys who really has pioneered this field and had the vision for a long time that has helped bring us to where we are today. So please welcome Dr. Michael West.
I feel like President Trump or whatever, they come out and they go, you know, like I know all you guys, right? Yeah, so thank you. You know, it's a pleasure to be here at this event. You know, we have a mutual abhorrence of aging and death, and I just don't run into a crowd like this every day, so it's a pleasure to be here. I'm going to talk to you about Ajax, not really. But we are a public company, and so I have to read this. No, I'm not going to read this. I'll be making forward-looking statements that have associated risks, so see our SEC filings for more detailed information about the company. Okay, that's that. All right, so Ajax is a biotech company, and we're focused on aging, the core biology of aging. And an important aspect of that is age-related degenerative disease. The second bullet point I have here is we have a technology we're diligently working on for what we call age reprogramming. I call it ITR, and we're doing that through a subsidiary called Reverse Bioengineering. You know, I'm not going to talk about our company, really. I was thinking about this, and I thought, you know, I'd rather talk to you kind of heartfelt about what I see going on the next 15 years in the field from the point of view of a scientist who 40 years ago came to, you know, something happened to me 40 years ago. Jim was talking about sort of spiritual things, and I'm a materialist, really. But something happened to me 40 years ago. Over a beer, I'll tell you, some of you about it if you want to hear about it. But I knew that my life was going to be spent trying to tackle the problem of defeating death itself. I rarely say that because, you know, I'm in the biotech community and I'm a card-carrying gerontologist, and that used to be, in years past, you'd even say anything halfway like that, people would cross themselves and tell you to leave the room. So I'm going to talk to you about what I was going to say I think is going to happen. I know it's going to happen. I'm certain it's going to happen. So let me start by talking about what is not radical life extension. These are survivorship curves. You know, we see them all the time in the aging literature. So, you know, the number of people alive, or animals alive, or fruit flies alive, or whatever, over time, the blue being the treated animals. That's not radical life extension, right? Even this isn't radical life extension, though useful. Extending the lifespan of a human or an animal 30% could make a big difference. It's important. It's going to happen. Rapamycin, metformin, and drugs you haven't yet heard about are going to extend human healthy longevity somewhat. That's not radical life extension. This is radical life extension, right? Now, the question is there, let's get serious, is there a biological basis to believe that such a thing is possible? Not only is there a basis to believe it, it's an actual fact now. And most card-carrying gerontologists know it. You'll hear it alluded to during this conference. And I'm going to quickly walk you through it. So Jim mentioned, Stroll mentioned part of this. Basic birds and bees. All of you, were made from a fertilized egg cell, and the reproductive cells that made you were made by parents before them, there's this lineage of cells that made us that have been proliferating before the Precambrian, in the Precambrian era, over 600 million years ago, since the dawn of life on Earth. Though the cells that made us have no dead ancestors, okay? That's the red line. It's called the germ line. Jim mentioned this. So, but the difference is the cells that you see branching out here at the bottom, they're the cells that make up our bodies. They're called somatic cells. They don't do that. They age and die within a few decades, whereas the cells that made us have been proliferating for billions of years. Now, if you're like me, on a Sherlock Holmes search to understand aging and intervene in it in our lifetime, you'd pay attention to this, okay? That's what we did. By the way, a lot of scientists, I won't have time to name tonight, were part of this quest. So the goal was immortality transfer. Could we learn the basic biology of the immortal substructure of life and transfer it into these cells we call somatic cells that are our loved ones, our moms and our dads and our kids and our parents and our loved ones? Immortality transfer, is that doable, and how would we do it? Okay, quickly, and I want to get into a lot of the science here. The first thing that happened back in the 90s at Geron, Bill Andrews is here, we'll talk about telomeres. We figured out the first step of what happens that makes you and I die and how we could intervene in it. The cells that branch off from that sperm and egg that make the body, as opposed to this reproductive lineage, become mortal. They lose the ability to replicate forever, and what we proved years ago at Geron and with our collaborators is that the clock is in the ends of the DNA strand. Lit up here, can you see like little automobile headlights? Those are called telomeres, end parts, that means in Greek, telos, meros. And there are little graphs showing how telomeres shorten. Every day, that clock is ticking away in many of the cells in your body. And so your cells in your body have a finite capacity to replicate. It's called the Hayflick limit. So cells divide, as you can see in this little video, and on the right is Hayflick's famous curve showing how cells age. But they can immortalize. We knew that. Scientists said, well, yeah, but we will never understand that either. Well, it turned out to be quite simple. To cut a long story short, we found the telomerase gene at Geron and put it into cells that age. And here you can see Hayflick's famous cells, young cells on the left, cells that have run out of lifespan on the right. Let me skip over those. So here's telomerase is this blob here, and it's an enzyme, and it adds DNA onto the DNA strands, extending the telomere, rewinding the clock, or however you want to, whatever metaphor you want to use. And if it's expressed, cells don't age. So on the right are cells with the telomerase gene turned on or added in, whatever, and they're like the Energizer Bunny. They just go, 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 go. They don't become cancer either. So the first fundamental step in understanding aging came in at the close of the millennium. I think this paper was published, 97, 98. Then came reprogramming. Dolly the sheep was cloned. I walked away from Geron to work in cloning, nuclear transfer. Some of you may remember the controversies about nuclear transfer, especially with human cells. And the nut and bolt of that is that the egg cell was like a time machine. You put a cell from an aged animal or an aged human into the egg cell, and we knew that something in the egg cell could take that cell back in time and literally reverse aging. Could we figure that out? Wow, that'd be cool. So it's been figured out. There, as you can see this arrow, we now, in fact, it's kind of an old hat, probably done in high school, science fair. Well, my son did it as a high school science fair project. We took some of my cells And I think the teacher knew I helped a little bit and didn't give an honorable mention. He took my cells, reprogrammed them using these reprogramming factors, reversed the aging of my cells back to the very beginning of life, and then turned them to the little baby Mike West beating heart muscle. And I should probably have shown you that. It's quite impressive. And neurons and things, and yeah, he got honorable mention. Why? As miraculous as that sounds, that's kind of old hat now. Did you guys even know that science can do that? A lot of people don't know that. Okay, so I don't want to walk you through a lot of boring science, but on the left is how we measure the age of cells. Can you see that kind of smear, that blob on the left, and then it goes down, then it goes back up? That was the prettiest picture I've ever seen in my life, other than my kids' pictures. That is the reversal of the aging of human cells. And we know it's real reversal of aging. In the middle is third party. Steve Horvath did a study with the methylation clock. It's an entirely different way of measuring the age of cells, a very accurate clock. And you can see IPS is back, all the way back to where ES cells, embryonic stem cells, are at the very beginning of life. So on the right, this is how scientists think about all this. You see the cells at the very top. They're the immortal germline. They fall down this inclined plane and become different kinds of cells, and you and me and our loved ones, blood cells and nerve cells and so on. We knew we had the technology of reversing aging all the way back to the beginning of life, to pluripotency it's called. The question then became, yeah, but we don't want to just turn you into a blob of unformed pluripotent cells. You would just like dissolve into a little puddle. That's going too far. Could we reverse the aging of cells, not just in the laboratory dish, but in a human being, back in time to reverse aging in a human being? Well, what's interesting about this is that there are animals like this salamander you see in this little video that is stuck in as a blob of cells, but as an intermediate stage of development, like what we are early in our development as a fetus or a newborn human being. And in that state, you've probably seen this, not in your backyard, but on TV or something. You cut a limb off these animals, and you see the little diagram there on the right? It grows back flawlessly. How could an animal, and by the way, you do that to the eye, the heart, the brain, it's throughout the body. How could that happen? Well, these animals are simply never fully matured, okay? So the idea was born for age reversal back to that regenerative state in a human being. If we could do that and combine it with the extension of telomeres, I know, in the next 15 years or so, you're going to be hearing about this being played out in real time in the world. And my belief is it's going to change the world in a remarkable way, maybe more than significant revolutions like the internet and computers and cell phones and so on. This is a truly powerful and remarkable technology. It's beyond description. The implications for all of medicine, from neurology to cardiology, people that have a heart attack, it scars over, your heart can't regenerate. It can regenerate early in your life, though. And so how can we accomplish this? Well, there are numerous papers. It's commonly called partial reprogramming. It's, I think, a weak term. When my kids, when they were young, they did like all your kids. Are we there yet, right? We're going on a trip. And I started saying, we're part of halfway there. And they would like, what? What's that mean? Part of halfway there. Or half of partway there, whatever I said. And partial reprogramming, what does that mean? I call it induced tissue regeneration, ITR. I don't know what the word is going to be. Induced age reversal, I don't know. The concept is we need to very carefully design this. Why? Because like telomerase and its role in cancer, this regenerative phenotype is commonly used in cancer as well. So the application of this new technology to humans, we've got to be careful that we don't induce cancer. So, and that's going to be the FDA's #1 concern, I'm sure. So what we've done is we've taken this to a new level. We have a precise way of regulating reprogramming. And for those of you that have a technical bent, I'll just quickly try to explain it. If we put these genes in a gene therapy vector, like I'm showing you here, it's called adeno-associated virus, and use you see these little bar graphs? The embryonic fetal transition is the target we want to hit, but we don't want to go past. We want to take tissue, like your heart or your skin or your retina or whatever. We want to take it back to that we want to make you a salamander man. We want to make you regenerate in an immortal fashion. The goal is to hit the embryonic fetal transition. So we've got genes, the switches that control whether these genes, reprogramming genes, are on or off. We've got a gene that will sense when you hit that time point and then turn the vector off. So we can reprogram cells, we believe, safely to induce the regenerative phenotype to allow your heart to grow back after a heart attack. That's the goal. Or your midbrain, if you have Parkinson's and so on, without taking cells all the way back to the beginning of life to pluripotency or to significantly increase the risk of cancer. So that's precision reprogramming, which I believe is a really important next step. And, we were talking earlier this evening, some of us, about Kennedy and the Moonshot. President Kennedy said within a decade, we're going to land a man on the moon. Took a lot of people and a lot of engineers, but the key was it possible? Did we have the technology in hand to accomplish such an ambitious goal? And what I'm telling you is, with these technologies and with these new precise vectors to reprogram cells in the body safely without inducing cancer, I believe the tools are in hand to accomplish this. What would it mean? Have you ever seen these animals? I used to collect them as a kid. This is planaria. You cut it in half. The cut here is photoshopped, but the rest is real. This is a time-lapse video. Watch the top. The tail is regrowing a head and new eyes. That's regeneration in a very profound sense. These animals have regenerative potential, that second step that you and I turn off early in our development, and they have telomerase. So they can regenerate forever. The animals don't age. There is no aging in planaria or similar organisms that are immortal regenerators. So there are clockwork mechanisms in somatic cells, one of which is the switch to mortality, the loss of telomerase activity, telomeres shorten, the clock is ticking in many of the cells in your body. Some cells don't replicate, so they don't lose telomeres. Animals with regenerative potential and telomerase don't age. And vectors exist for the introduction of these reprogramming factors. We believe, we hope, will be safe in humans. And this will play out in the lifetime of many of you now living. Those are facts. Where all this will take us is unknown, but what I commonly tell people is we need, in a sense, a brave new world. Not the scary type in the novel necessarily, not at all. We need to be brave because we have loved ones we care for. And at least I do. I think many of you do. And the way I think of it as a gerontologist, if I saw one of my children or one of yours in a burning building and they needed to be rescued, I would risk my life to save them. I would be brave. I wouldn't go into a burning building otherwise. We need a new world because if you go into a nursing home today, I know there are people out there who say we shouldn't go there. We were barred from the tree of life, you know, in the book of Genesis. Whatever, world population explosion, whatever their objections are, we shouldn't go there. Go into a nursing home. See grandma, grandpa with Alzheimer's not recognizing their loved ones anymore, or in the pain of osteoarthritis. We can do better. We need a new world. So my belief is that this technology can be applied compassionately. It will come. When is a matter of, well, President Kennedy mobilized an enormous effort to accomplish the goal of a lunar landing. Jeff Bezos, you probably have heard, recently was a member of a group, I guess, that committed $3 billion based on the technology I just described to you. So there is growing recognition in the scientific and business community that finally, placed in the hands of mankind, are technologies that will lead to radical life extension as opposed to modest impacts on aging. There's a quote in a well-known book on regeneration: if there were no regeneration, there'd be no life. The germline would cease to produce humans. If everything regenerated, there'd be no death. And with that, I would thank you very much for your attention. Thank you.